Today's episode is all about excitation, also known as excitatory neurons. Excitatory neurons are a part of this push-pull system of excitatory and inhibitory neurons. Of course, excitation is activation, or start, or go signals. We've recently covered a few episodes on the inhibitory side, the inactivating, or stop signals. Those are episodes. Episode 76, Parvalbumin, interneurons, and the autistic phenotype. Remember, Parvalbumin interneurons are the most common inhibitory type of neuron. These are very fast-acting, and often the autistic phenotype has the deficiency or a lack of in key areas such as the anterior insula, the ACC, the dorsolateral prefrontal cortex, and the thalamic reticular nucleus. Episode 82, Sonic Hedgehog and Inhibitory Neurons in Autism. Episode 83, Thalamic Reticular Nucleus, or the TRN, Sensory Gating and Autism. In addition, we have episodes 4 and 5, a two-part series about just general excitation and inhibitory. The reason is this. The recent episodes are zooming in to cover the specific cells and their roles. Within the autistic phenotype, there is a significant battle between this excitation and inhibition imbalance. Excitatory neurons are primarily pyramidal neurons, and they release glutamate, the brain's main excitatory neurotransmitter, to activate postsynaptic neurons via AMPA and NMDA receptors. Remember the spatial relationship of AMPA and NMDA on the postsynaptic area of the synaptic cleft. These neurons drive neural activity and promote information transmission and are critical for sensory, cognitive, and motor functions. Remember, overall, the human central nervous system, the brain, has three main types of neurons: sensory neurons, interneurons, and motor neurons. In the autistic phenotype, excessive excitatory activity contributes to an elevated excitation-inhibition ratio, and this leads to sensory hypersensitivity, of course, cognitive deficits. The primary one here is a lack of adaptive responses, not being able to adapt properly or with a large toolbox, if you will, in the social world. And also repetitive behaviors, this black and white, these rigid thinking, the fixated interests that are so-called abnormal in intensity or focus. This EI imbalance contributes to these things. Overall, I can't comment on if the autistic phenotype has an abundance of excitatory neurons and or a lack of inhibitory neurons. Now, I know this. The data are impressive on this. And based off of the Reza Shadmir episode about the cerebellum and the Parvalbumin data, there are a lack of inhibitory neurons, especially the Parvalbumin interneuron and the Purkinje cells of the cerebellum. There are, indeed, a lack of those. I am unsure if it's enough, if that's adequate enough to cause this imbalance. Because another region of interest is this, that hypoconnectivity with distal connections of the brain, such as the superior colliculus to the frontal eye fields or the dorsolateral prefrontal cortex. There's distal connections in the human brain, and the autistic phenotype struggles with that as well. So the main cell type here in the excitatory neurons is pyramidal neurons. This is characterized by its shape, large triangular, and extensive dendritic trees, and long axons for long-range connectivity. Now, if you are wondering why it is called pyramidal neurons, it's because it's nothing creative. It's nothing special here. They just look like a pyramid. These excitatory neurons act on those AMPA receptors and NMDA receptors. This is glutamate. And those are both ionotropic receptor types, very fast. They're faster than metabotropic, which is glutamate receptors mGlu-R 1 through 5. These are more modulatory. Now, you might ask, and you ought to ask, what are these excitatory neurons? How are they different than the excitatory neurons of neuromodulators, such as acetylcholine and dopamine? Remember, dopamine has 2 receptors that are excitatory and 3 receptors that are inhibitory. And of course, we have excitatory dopamine and inhibitory dopamine. These are a little bit different. Glutamate transmits information in a fast, like a light switch type of activity here. It's instant. Neuromodulators determine the processing. They modulate the excitability to keep firing or keep paying attention. And these happen seconds to minutes versus the 1 millisecond to 10 milliseconds of the AMPA and NMDA receptors. The modulators are like a volume control, or if you have a adjustable light switch, we can determine the lighting. The modulators are state setting, determines your brain state, and determines attention, motivation, and salience. So really the process is these excitatory and inhibitory neurons acting fast, within 1 to 10 milliseconds. And then neuromodulators are on the scene, within 10 to 30 seconds. And then, if we really want to get into it, hormones are activated about 30 seconds on scene. Okay, with the pyramidal neurons, there are little subtypes here. Layer 2 and 3 pyramidal neurons facilitate cortical connectivity. So for example, visual cortex 1 to the dorsolateral prefrontal cortex. It's one of the most distal connections in the brain, from the very back of the brain, all the way to the front of the top. That's the dorsolateral part. And then layer 5 and 6 pyramidal neurons. And these project to subcortical structures, such as the thalamus, the striatum, thalamic reticular nucleus, and so forth. Hippocampal pyramidal neurons, or NCA1 and NCA3, very similar to the inhibitory neurons that we discussed. This is critical for memory. And we also have granule cells. These are dentate gyrus of the hippocampus cerebellar cortex, and excitatory inputs to inhibitory neurons. Remember, excitatory neurons and inhibitory neurons can act on each other. The synaptic properties here, the high synaptic plasticity via NMDA dependent long-term potentiation. These are huge for learning. We'll cover learning shortly. And long-term depression. This is modulated by BDNF. We'll talk a little bit about brain-derived nootropic factor in a bit. Some brain regions associated with the pyramidal neurons. The cerebral cortex, of course. Now pay attention to this, because these are very similar. They are identical to the inhibitory neuron episode. So the dorsolateral prefrontal cortex, the ventromedial prefrontal cortex, the orbital frontal cortex, the anterior cingulate cortex. Those are all very popular in the podcast. We've discussed each one of those regions. And the sensory cortices, primarily the visual cortex 1 and the subsequent layers. Somatosensory cortex, remember S1. And the primary auditory cortex, A1. We've discussed V1, S1, and A1 in the inhibitory neuron episode. The insular cortex as well, the anterior insula especially, and the posterior insula. The insula is a very popular region of mine and of the podcast. Very important. It's very privileged. And the temporal cortex, especially the superior temporal gyrus. Remember the decoding the brain, how reading works in autism and dyslexia. We talked about the STG in quite detail. Inferior temporal cortex, huge for object recognition. And then the hippocampus, the CA1 and CA3, and the dentite gyrus. And in the amygdala, the basolateral amygdala is heavily populated with excitatory neurons, and the cortical amygdala, more for social behavior. Now, hard stop here. The days of saying this region is responsible for this region equals this action, this behavior, this thought, this feeling, etc., are over. Neuroscience has switched off of that for great reason. It's more about neurocorrelates, networks, pathways, connections, and so forth. Oftentimes even, such as this amygdala example that we're using. Yes, it's fear, but it's also the opposite of that. It is not about the region. It's about the network and connections. The amygdala is often the biggest misnomer, especially with this so-called fear center. That term oversimplifies its complex role in emotional processing and does a disservice to its broader functionality. And this is what we're going to cover right now. It is a huge misnomer. While it's true that the amygdala is critical in detecting and responding to threats, rapid processing sensory cues to trigger fear responses, like an increased heart rate or freezing. It is not solely detecting fear. This misnomer stems from early neuroscience research, particularly studies on fear conditioning, which highlight its role in associating stimuli with danger. However, the amygdala's involvement spans a wide range of emotional and cognitive processing, making the so-called fear center tag misleading. It's more accurate to view the amygdala as an emotional hub. Hub is a very good term, and it's evaluating the significance of experiences and coordinating the brain's response to both positive and negative stimuli, with fear being just one piece of that puzzle. And remember everything we've discussed recently about brain waves, sometimes called frequencies or oscillations. The amygdala's functions are very diverse, and they encompass emotional processing, memory enhancement, reward evaluation, and social behavior. The amygdala assesses the emotional weight of stimuli, whether it's a joy for something you like, or you want, you need, you love, or the thrill of a potential reward, or some sort of social connection. And this is integrating sensory input with internal states, like mood or hunger. For instance, it activates during rewarding experiences, such as eating or social bonding, and is interacting with the brain's reward system, including the ventral striatum, which is in large part the nucleus accumbens. The nucleus accumbens is the ventral striatum, and it's a very large brain region. And dopamine pathways. The amygdala strengthens memories tied to significant events, both positive and negative. And it makes these moments, like remembering a pleasant first experience of a certain stimuli, or a near-miss accident, more vivid. It kind of stamps these. In addition, the amygdala decodes social cues, like facial expressions, to gauge trust or affection. And it also collaborates with regions like the prefrontal cortex and hippocampus to regulate emotions and contextualize the experience. One thing that really throws this misnomer, gives it strength, I should say, is the so-called low road versus high road. And this is very true. If you see a snake and you instantly jump back, you're startled, you're alert, so forth. That's the amygdala kind of lighting up because the visual sensory makes a quick stop at the amygdala for these things. And that's the low road. But then eventually you think, oh, not a venomous snake. That's not a copperhead. That's a water snake. And then that's the high road, kind of providing context based off of memory, based off of the living organism, really, to say, no need for startle, no need for this fear. And by the way, a copperhead and water snakes are very similar. They have this black shape. The copperhead, the shape is in the shape of a Hershey kiss, a triangle. And the water snake, it's more of a tornado, so it's an upside down triangle. Or even sticking to the theme of the episode, we could call it a pyramid, based off of the pyramidal neuron. Hershey kiss, copperhead. Tornado, water snake. Okay, we were talking about brain regions, and I need to pivot back. It's hard sometimes. So we finished up the amygdala there. And also the striatum, the caudate, and the putamen. So that's the dorsal striatum. Remember the basal ganglia, the input centers of the basal ganglia. That's our go, no-go area. It's modulating response types. And also the ventral striatum that we just mentioned, the nucleus accumbens. Pyramidal neurons are dense in these three regions. The thalamus, of course, the thalamus. Relay neurons and the lateral geniculate nucleus. So remember vision, you should know that's vision. The ventral basal complex, which is VB. We'll talk more about that momentarily. And the somatosensory. In addition, in the thalamus, the medial geniculate nucleus. This is auditory. And the medial dorsal nucleus for cognitive and emotional processing. And the cerebellum for those granule cells. And this is the excitatory inputs to Purkinje cells. Okay, I know that's a lot of nomenclature, a lot of dense neuroscience and brain regions. But they're all very relevant here. They're all important if you want to understand certain phenotypes, like the autistic phenotype. So the receptors, let's touch a little bit more on the receptors. AMPA. And these are very fast excitatory transmissions, such as I was touched. And they use sodium, potassium. And they are activated 1 to 5 milliseconds. The NMDA receptors use sodium, potassium, plus the calcium. These are critical for synaptic plasticity, the long-term potentiation, and long-term depression. Remembering the texture of what was touched. AMPA will say, I was touched. NMDA will say, oh, I recognize that texture. I know what touched me. And NMDA are slower. They open at roughly 10 milliseconds and stays open for longer, 50 to 200 milliseconds. And also the M-glutamate receptors, M-glu, GLU receptors, 1 through 5. This modulates excitability and plasticity via G-protein coupled signaling. These are metabotropic receptors. They are slower than the two previously mentioned ionotropic receptors. And some interactions here. These excitability receptors receive inhibitory inputs from GABAergic interneurons, the Parvalbumin interneuron, the somatostatin inhibitory neuron, and the vasoactive intestinal peptide, the calbindin, and the calretenin, and the Purkinje cells. All of those inhibitory neurons that we previously discussed. This is via GABA A, B, and C receptors. A general role here for these excitatory neurons and receptors is to drive neural activity for sensory processing, especially visual cortex 1, somatosensory cortex 1, and auditory cortex 1, and for cognitive functions, especially in the dorsolateral prefrontal cortex. For emotional regulation, some regions of interest here are the anterior insula and the amygdala. And the motor control, remember the striatum and the cerebellum. They also facilitate synaptic plasticity for learning and memory. Huge. And they generate gamma oscillations from the 30 to 80 hertz range. Remember, these are activating, telling us to go, in coordination with Parvalbumin interneurons. In the autistic phenotype, there's an excessive excitability, excitatory activity in the sensory cortexes, especially the visual cortex 1, the somatosensory cortex, and the auditory cortex. We've covered this in great detail. This is due to brain-derived nootropic factor-driven synaptic enhancements and reduced inhibitory control. And this elevates that gamma power, and it drives sensory hypersensitivity. This impaired EI imbalance in the dorsolateral prefrontal cortex and the anterior insula reduces gamma synchrony and contributes to cognitive and social deficits, especially, remember, those distal connections. It's a big mismatch, a timing mismatch. Overactive corticostriatal thalamic cortex circuits, especially the orbital frontal cortex and the striatum, amplify repetitive behaviors. Remember, the basal ganglia is involved there. So what is the neurobiology and the purpose, this evolutionary aspects of excitatory neurons? Information transmission is huge. Synaptic plasticity, these so-called network activations. This is now and the future of neuroscience, networks. And the thalamic reticular nucleus interaction. So excitatory inputs from cortical layer, visual cortex 1 pyramidal neurons to the thalamic reticular nucleus modulate sensory gating and oscillatory activity. It has to be a healthy balance here. Just think about this push-pull tactic. So what's the difference between typical and the autistic phenotype with the excitatory neuron functions? In large part, it's the get up and go, the activation versus inactivation, orchestrating which regions to turn on, in addition with those neuromodulatory cells. It's all a network here, an orchestra, for modulating movements and understanding, processing, understanding your environment and what you're going to do when you encounter these things in the environment. And also with the brain waves. Remember the different brain waves here. Each wave range has different functions. That has a lot to do with brain states, bodily states. The autistic function here. Let's start with the sensory cortices. V1, S1, and A1. So the hyperactivity. You can all understand the hyperactivity. Excessive excitatory activity due to the enhanced pyramidal neuron firing and the reduced Parvalbumin and somatostatin inhibition. This must be a healthy balance You have to activate and inactivate. Activation and inactivation happen simultaneously. And elevated gamma power. Increased gamma power, especially in that 30 to 80 hertz, and V1, S1, and A1 is very much a huge thing with the autistic phenotype. This hijacks the sensory processing. What are you paying attention to in the environment or even within yourself? This is huge. This is why the autistic phenotype has that attention to detail. Remember the tree bark example and the visual thinking. I believe episode 2 of visual thinking. If you think about a tree, do you see the tree or do you see specific details about different trees? And in large part, this is a source of the visual thinking as well. This imbalance of excitatory and inhibitory, especially at those early connections. We get stuck. There's so much to process right there. The rate of information coming in is so fast. But then those distal connections are huge. This so-called higher order regions of the prefrontal cortex and so forth. Reduced activity and synchrony because of this imbalance of EI. I can't overstate this enough. If you go from point A to point B, and let's say there are 5 stops that you have to make to reach your destination. The autistic phenotype, let's say the first 2 stops, we get there fast. So fast versus the typical. But the final destination, achieving and reaching that final destination, the typicals will get there faster because there's this connection. It's about the overall point A to point B. How we get there differs. For the autistic phenotype, it's skewed to the early connections. We get there so fast. But the subsequent stops, especially, to the prefrontal cortex, that's a major region for how we evolve and executive functioning. We get there slow. And just think about neuroplasticity and how we store memories and expectations. How do you think this is going to build up with experience and over a lifespan? Do you think this is going to be stored as pleasant memories? Because mostly what we're talking about is the social environment. Internally, all good, no problems. It's actually a huge benefit here. Remember, the biology that gives us autism allows us to be comfortable within ourselves. That's a huge source, or the top source, of the accelerated learning. We give ourselves a chance. There's no internal conflict or barriers. So some roles in learning with the excitatory neurons. In large part, it has to do with the synaptic plasticity. Remember, the brain doesn't want to work. It just wants to respond. It responds based on what it knows, what kind of connections are being made and strengthened within each of us. And that will be privileged. The brain doesn't want to utilize other connections because that's work. You might think about how change is hard for the human being, and indeed it's true. But think about it from the synaptic view. Go inside the brain and the central nervous system and view it from that perspective. This will allow it to make better sense. Whenever we repeat things, the so-called cells that fire together wire together, and the strengthening and the growth of synaptic connections. That's the preferred choice for the living organism. And then if you add in the brain waves, if it's one-trial learning or gamma or high gamma, it's strengthening these connections faster. That's why if you almost get into an accident, a near-miss accident, that's really heightened and it stamps your memory much more than the previous time you drove to work and there was no accident or close call. The role of the brain-derived nootropic factor, BDNF, enhances NMDA receptor function. This is in large part the driving mechanism behind this. BDNF is so huge, especially in the critical period, whenever children can just learn multiple languages, like passively, with no problems, no disruptions. In large part, BDNF is modulating that. It allows it to happen. And if you think about the autistic phenotype, altered plasticity, there's this so-called hyperplasticity because of the excessive BDNF driven long-term potentiation, especially in V1, S1, and A1. This enhances sensory learning, leading to superior detailed-oriented learning, like visual pattern recognition, but an overall reduced flexibility. In the hippocampus, overactive CA1, CA3 pyramidal neurons, due to that reduced somatostatin and Parvalbumin inhibition, impair pattern separation. And this creates a rigid memory encoding. Remember, these are all the same areas that we discussed in the inhibition episodes. Dorsolateral prefrontal cortex impairment, a reduced EI imbalance in this brain region impairs working memory and associative learning. And this contributes to that cognitive inflexibility as well. The dysfunction in the thalamic reticular nucleus, an impairment of Parvalbumin inhibition in the visual and somatosensory and auditory sectors, allows for excessive sensory input back up to the cortex. Remember the thalamic cortical pathway, enhancing sensory learning but overwhelming cognitive circuits. And the limbic sector of the TRN has a deficit because of the reduced dorsolateral prefrontal cortex excitatory activity. This impairs attention and dependent learning. In the autistic phenotype, some strengths here is enhanced sensory learning in those early visual, somatosensory, and auditory networks. This is superior attention to detail. Some challenges, though, are impaired creating inflexible, the lack of adaptive responses, and leads to context-dependent learning. Just tell us. Just be specific type of thing. So also think about repetitive behaviors here and social deficits as well. And OCD-like behaviors. OCD and the autistic phenotype share some very similar neural circuitry. Overactive excitatory inputs in the corticostriatal thalamic cortex circuit. Due to the reduced inhibition control, this amplifies habitual learning and drives repetitive patterns. Something as similar as tics Tourette's is overexcitation. We've talked about stereotypy. It's a misnomer as well. It's defined, if you look up the definition of stereotypy or stereotypy, it will say purposeless behavior. But there is very much a purpose of why these repetitive behaviors and stimming and OCD-like tendencies, tics Tourette's, there's a purpose, there's a reason. These things are happening. Humans, well, just define things like so loosely without even considering. So let's wrap up with the push-pull analogy here. The go signal explanation of this push-pull. Excitatory neurons act as the brain's go signal, and this drives neural activity to initiate and propagate information processing. Glutamate releases via AMPA or NMDA receptors, and this depolarizes postsynaptic neurons, while increasing their firing probability and promoting 1. Sensory processing, cognitive processing, emotional processing, and motor output. V1, S1, and A1, so visual, tactile, that touch, and auditory stimuli. Cognitive processing, especially in the prefrontal cortex, Pyramidal neuron supports attention and working memory. The emotional processing centers. The basolateral amygdala is huge here, and the anterior insula drive emotional responses. What are you paying attention to? There's a reason why the insula is part of the default mode network and the salience network. Motor output. The dorsal striatum, and even the ventral striatum with reward learning. And the cerebellar. These are motor movements. Remember, the dorsal striatum is our input area, and this will regulate and determine action selection. In typical brains, this go signal is balanced, acting as a stop-slow signal. You have limitations on how fast you can go. In the autistic phenotype, that excessive go signal and the BDNF-driven excitatory enhancements and the reduced inhibition amplify everything. The 2003 paper from Rubinstein and Mike Merzenich, who studies neuroplasticity, is a good foundation to start with this EI imbalance. An EI push-pull analogy here is the push, the excitation. Pyramidal neurons in the cortex and the thalamus push neural activity forward, driving sensory, cognitive, and emotional processing. The pull system here, however, the inhibition, those inhibitory cells and the Purkinje cells pull this back. It's a brake system. Collectively, they're working as bumpers on, let's say, a bowling alley. And this is all one, depending on the context, and living organism dependent, depending on what the living organism is thinking, wanting, liking, trying to accomplish, and so forth. I have something exciting. I want to introduce a product unlike any other product available. A highlight is the product from Daylight Computer Company created their product based on these factors. The Daylight computer is completely blue light free. It has no flicker. Short wavelength flicker is extremely harmful for our eyes and downstream biology. Light flicker is constantly turning our central nervous system on and off. Essentially, it is like going to a light switch and repeatedly turning it on and off. The problem is, blue light and LED light does this, and it is so rapid you cannot even perceive this in real time. The Daylight computer is the lowest stimulation and foremost for sensory sensitive users. It is no question that the alternative product, especially when used at night, do not address or consider this in their product. It is so toxic to human biology. Big tech corporations have patents on how their short wavelength implicate the human nervous system. And a bonus, despite Daylight computer not having backlight, it is very functional for outdoor use. And of course, increased sunlight is always preferred. I am happy to offer a discount for the Daylight computer. You can use the code AUTISM for a $50 off discount. Again, use the code AUTISM and the discount code for $50 off. See the link in the show notes to Daylight Computer Company, or just give it a quick search in your internet browser. Use the code AUTISM for $50 off. I would like to mention Chroma, lights designed for humans. Chroma, a Seattle-based innovator founded by ex-NASA and Air Force engineer Michael Shapiro, is on a mission to enhance physical and mental health with purpose-built devices. unlocking peak human health, cognitive function, and performance. Shapiro launched Chroma to restore the natural light lost to screens and indoor living, and delivers faster recovery, sharper minds, and better sleep. Their products hit hard. The Iron Forge speeds muscle repair with red and near-infrared light, while the Skylight mimics sunlight to boost sleep and energy. A standout, the Forge lamp, is a portable gem that fires 660 nanometer and 850 nanometer light to energize mitochondria and heal tissues on the go. Remember the 4 red light chromophores on cytochrome C oxidase, frequently talked about in the podcast. This is why. Their products are built with military-grade durability that have lasting impact. Chroma's tech fuses precision and power. Gallium titride power supplies, smaller, cooler, and stronger than silicone, provide flicker-free light that's easy on the eyes. High-powered LEDs target key wavelengths for skin, tissue, and cellular health, with smart heat management for the lasting impact. Every design decision Chroma makes serves a purpose: to create devices that are precise, durable, and effective for improving human life. Remember, humans use different wavelengths of light for different functions of life. Remember when I ask, what do you think light is? Chroma designs with our biology in mind, from sleep aids and wound healing to mitochondrial energy, full body lights and blue light blocking glasses. They are US made and Chroma ships globally and accepts FSA and HSA payments. Use Autism at checkout for a 10% off discount. That's Autism at checkout for a 10% off discount. If you are listening to the podcast, listening to the episode, please feel free to leave a review or rating. In podcasting, reviews, ratings, and downloads are huge, and I very much appreciate your feedback. You can contact me on X at RPS47586. We can discuss anything and everything about autism. I very much appreciate your comments and your interactions on X. You can check out the YouTube page for all the videos, full-length videos, shorts, and clips. You can email me, info.fromthespectrum@gmail.com. Thank you for listening to From the Spectrum podcast.